Good morning, guys. We are going to start Lecture 4, Chapter 4, and this is carbon and its molecular importance in life, or its atomical purpose in life, if you will. So let's get started on that. We can start applying some of the chemistry we talked about, right? So carbon is really, really important in life itself. This picture here is a picture of a cicada shell. You've probably seen them, right? Especially on those years where you get lots of cicadas that come out in the summertime. This whole shell is carbon-based. It's made up of a sugar that is called chitin, right? And chitin is what makes up the exoskeleton of insects. And it is a carbon-based compound. And the beauty of carbon is its ability to make many chemical bonds, and hence why it's been chosen, if you will, for life, right? Or for the diversity of compounds, particularly really large macromolecules that it can make. So living organisms consist mostly of carbon. Carbon is one of the major elements, an atom that is found in living organisms. Carbon is unparalleled by its ability to produce big, large, complex, branched, very diverse, if you will, structural molecules. It makes up DNA, proteins, lipids, fats, all of these things, RNA, are all carbon-based. So living matter is a large percentage of carbon. So organic chemistry, as many of you will take, or maybe some of you are taking, right, is the study of compounds that have carbon in them, right? So organic compounds range from very simple molecules to these gigantic, huge, enormous, or what we call colossal, very big compounds. Most organic compounds contain hydrogen. So most of the compounds we'll talk about are hydrocarbons or variations of hydrocarbons. They have a lot of carbon, not only carbon associated with them, but a bunch of hydrogens or protons associated with them. And so they've got an organic chemistry apparatus over here. It's a study of chemical reactions, right, of carbon, both synthesizing and breaking them down. We'll leave the organic chemistry to the organic chemists, and we're going to apply some of the basic chemistry that we went over to the importance of carbon and what carbon can do. So carbon has a valence of 4, and what that means is it has 4 valence electrons, right? Which is really, really great because those 4 electrons can participate in chemical reactions. So for example, here we got 2 carbons that are sharing 2 of the pairs of electrons here, right? And then we have hydrogens that are sharing the others, right, on each of the carbon atoms. And so if you fill in those valence electrons with electrons from other atoms, then their valence shell is filled and the carbon atoms are happy, right? So you got covalent bonding here. This particular type of molecule here, this 2 carbon-based molecule is nonpolar, right? And a lot of hydrocarbons, or almost all hydrocarbons, are nonpolar. And that's really, really important because they are very structural, right? These really large, let's use a different color than red. These really large carbon-based compounds are very structural in their basically job, right? Or their physiology. And not only are they structural, but they're hydrophobic, right? So they're hydrophobic, and that's important because their hydrophobicity means that they won't dissolve in water, and they form kind of the architecture, or what I like to think of as the scaffolding, the backbone of a lot of cellular structures, and therefore tissue structure, right? So carbon can bind to itself. It can form not only single bonds, but double bonds and triple bonds, or it can bond to other elements or atoms to form some very complicated and complex molecules. So let's look at this. This is what carbon, pure carbon, looks like when it's bonded to itself. It's kind of a black, coal-like compound. And of course, diamonds are all carbon, right? That have been compressed into a crystalline structure that you find in rings and whatnot. So here are some examples of hydrocarbons because they have hydrogen associated with them. So we got methane, right? We use methane a lot to burn in our heaters or our stoves. And this carbon-hydrogen bond, when you break it, releases a good amount of energy. And that energy is what is utilized, that we use what we burn in our heaters, for example, or our stoves to cook our food. Here's ethane. All right, ethane, ane. When you see ane on the end of the word, that means that basically you got two anes here, right? That means you have single bonded carbons are associated with either other atoms or carbons themselves. So methane and ethane, notice all the bonds are single bonded. When you see ene, like ethene here, all right, ene or ethylene is another way I learned it. E and E means you have a double bond between two carbons or more than one double bond in the molecule between carbons. So ethylene, ethyl means two, all right? So you got two carbons. Methyl means one, all right? So you can see ethane, two carbons that are single bonded. Ethene, two carbons that have a double bond. Methane, one carbon with four hydrogens. All four of these bonds can be broken to release energy. So those are some simple carbon compounds. Let's look at some others that are involved here with life. And if you remember, I told you earlier that about 96% or more of your body is made up of carbon, hydrogen, oxygen, and nitrogen. And these four elements play major roles in building large macromolecules that are going to make up the architecture and the backbone, if you will, of cellular structures. So the valence of carbon, as I mentioned, is 4. Nitrogen has 3 electrons that are single electrons in its outer shell, so it's got a valence of 3. Oxygen has 1, right? So, I'm sorry, 2, so it's a valence of 2, and hydrogen has 1, right? And so all of these can participate in either 1, 2, 3, or 4 chemical bonds. And so you can organize them in a whole bunch of different ways to form basically an infinite number of very large macromolecules. All right, so here's a couple of these. Right? Carbon can form multiple types of bonds we've already talked about. So here's carbon to oxygens. This is called, if you add nitrogen to it, this is called urea. So here's urea down here. All right, so this is a double bonded O here to 2 nitrogen groups. Right, this is how we carry all of our nitrogenous wastes in our bodies, for example, is through urea. So what urea looks like in a space filling model. Here is carbon dioxide, what carbon dioxide looks like. It's double carbons, double bonded on both sides to oxygen, very stable. Right, this is the waste product that we produce when we burn sugars, right? And that carbon dioxide can also be used to build sugars. So anything with carbon in it can be used to build really large molecules. So carbon can partner with any of those 4 atoms we just talked about to form very complicated and very large structures. All right, so molecular diversity. So you can see down here we got methyl, ethyl, propyl, all the way to decyl. All right, this is counting in numbers. So methyl's 1, ethyl's 2, propyl's 3, butyl's 4. Penta, which wasn't on your slide, I put it on here this morning. I missed it for whatever reason. Means 5, hexyl 6, and so forth. So those are 1 through 10, and those numbers represent the number of carbons that you find. Once you get past 10, then it just gets huge, right? So you can see propane here. All right, again, a very, very energetic molecule because of all of the hydrogens that you can break and produce energy by burning it. So propane, remember the ane means all single bonded. Ethane, single bonded again, ethane meaning 2. Non-ane would be 9, right? So and so forth. So you could use these prefixes to tell the number of carbons in a larger and larger and larger hydrocarbon molecule. Carbon chains can vary in length, and they can also vary in the way they look based on branching. So here's butane, and when I learned this, we call this isobutane, right? Because it looks different than the linear butane. There are 4 carbons. Here we have 3 carbons and then a 4th one branching off of it, and it's still a complete hydrocarbon, right? So butane, again, is you can burn it, right? It's got lots of hydrogen bonds to it that can release a lot of energy when they're broken. This is also called methyl propane, propane being 3 with a methyl group off of it, right, off of the branching. And you can add branching all over the place and make these really, really large and complicated. So there's lots of variation. Again, I'm not going to spend a lot of time going over the naming process and all of that. But again, I talked about enes and anes, right? So butene, 2 butene, 1 butene tells you where the double bond is, right? 3 butene would be over here. Right? And so you can number the carbons. When you add double bonds and you remove some hydrogens, right? So that each carbon still has 4 bonds around it, as you can see. When you remove some of the protons, you remove some of the energy, right? So energy is released. So that kind of tells you the double bond position. I like this picture down here is a long carbon chain, and it shows you double bonds. And what double bonds do is they kink your straight carbon backbone. So if you have all single bonds, you got a nice straight backbone. If you add double bonds, you kink it, right? Like fatty acids. When you have unsaturated fatty acids, they're going to have kinked tails, which means that they fill in more space. Their space filling model is going to be larger, fills in more airspace, if you will. And that becomes important when you start talking about oils versus lards and whatnot, which we'll talk about when we get to the macromolecules, which is the next chapter. You can also form rings with carbon. As you can see, you can form rings here. This is cyclohexane. Cyclo meaning it is a ring, right, of 6 carbons. And again, it's just simply a hydrocarbon. Over here, we got benzene, where we have double bonds, every other carbon. And these are solvents, right? Benzene's a solvent. Cyclohexane is a solvent often used when you're trying to dissolve hydrocarbons because they're hydrophobic, right? These ring structures are very stable and strong. And ring structures can become a major part of basically molecules that we find in the body, such as steroids. So, you know, down here we got cholesterol down here, and we'll talk about the importance of cholesterol because cholesterol is found in cell membranes, and it also is the basis of all steroids. For example, sex steroids like estrogen and testosterone. And the ring structure has a very specific three-dimensional shape and is very stable, right? And it'll fit a protein receptor very nicely so that it can stimulate responses in cells. And we're going to call those ligands later. So just give me a little foreshadowing of what we're going to kind of build upon and get into. But these ring structures become very important if you want to stabilize architecture, make it stronger, or if you want to form some of these communication molecules, which I call ligands, right? So these can become ligands or communication molecules because of their very specific shape. So as I mentioned, hydrocarbons are carbon chains that are saturated with hydrogens only, right? Many organic molecules such as fats have hydrocarbon components where it's just hydrogen and carbon. And as I mentioned, the hydrogen-carbon bond is very energetic. So it makes sense. Fats, if you've always learned, probably heard that most of the fat in your body, like the fat found under your skin and around your organs, that what we call triglycerides, are high-energy molecules. And they're high-energy molecules because their long tails are long backbones of carbon with lots of hydrogens. The more hydrogens I add to the carbon chain, the more energy that I can store in it. So it's a very nice way for us to store energy, chemical energy, that we can utilize later. So as I mentioned, they can release large amounts of carbon. So you can see the longer and longer I make this carbon chain, the more hydrogens I add to it. Each bond releases energy when I break it. The more hydrogen bonds I have, the more energy that is reserved in those hydrocarbons. Most of your triglycerides, there'll be 3 right? 3 long chains of carbon can hold about 126 ATP molecules. And ATP, as you probably heard, is our energy molecule. So it's worth that much ATP, which is a great way to store energy for later on down the road. So here is one of these, what we're going to call a fatty acid, right? That's what this is down here. This is a fatty acid. Okay, I'm going to get rid of that one because it's not very bright. So this is a fatty acid down here. All right, the triglycerides have 3 of these attached to them. And you can see it's a really long hydrocarbon. On the end of it, we have the acidic portion because this hydroxyl group here, this OH, can give its hydrogen when you're in solution. And remember, that's what the definition of an acid is, to give protons to solution. So that's why it's called a fatty acid, because it's going to be associated with fats. This type of group here on the end of the molecule is called a carboxylic acid or carboxyl group, right? Is what you learn in organic chemistry or in chemistry maybe. And I'll show you the functional groups here in a minute. So carboxyl groups give off hydrogens. So here is what a triglyceride looks like. So these are, you can see there are 3 of them. These are the 3 long fatty acids. Right? And they are attached to 3 carbons up here. That is called a glycerol molecule. That's what the 3 carbons are. It's a type of alcohol. So glyceride means triglyceride means a glycerol molecule with 3 fatty acids on it. Then triglycerides, right, are the basis of our, what we call fat that's found under our skins, our cellulite, right, or what we call adipose tissue. So this is what adipose cells, these are individual adipose cells. All right, so this is an adipose cell here, actually. This whole thing is an adipose cell, sorry. Right, and I'm going to draw this in so you can see it, but right here is kind of a cell membrane. All right, you got this is under an electron microscope. Many of you might not be familiar with that. On the other side out here, this stuff out here is extracellular space. This is the inside of a piece of the cell, and this is the nucleus right here. You can see the nucleus. And then you can see these individual droplets, if you will, of triglycerides. And they're made up of these, right? These fat droplets inside of this particular adipose cell. Okay, so functional groups. Here are some of the major functional groups. There are really 7 major ones in biochemistry that are associated with our organic molecules that participate in biochemistry and cellular biology. And so what I would do immediately is become really familiar with them. Memorize them, know what they look like, so that you can recognize them on exams. You can recognize these groups on exams. And these groups, these functional groups, become very important because they help determine what the molecules or compounds we're going to talk about look like, and therefore what their function is, right? So I just kind of started that, right? A hydrocarbon is energetic. It has lots of energy in it because of the carbon-hydrogen bond. The more carbons you have, the more hydrogens you add, the more energy that that molecule can hold. All right, so all of these groups here have really important jobs when we start talking about them in biology, particularly when we look at the individual macromolecules that we're going to talk about in the next chapter. So these are the most common ones. So you got hydroxyl groups up here, right? So here's ethanol. A lot of the alcohols, when you see the O, L on the end there, the OL here on ethanol tells you it's an alcohol. Alcohols have hydroxyl groups on them. Like this is ethanol because it's 2 carbons with the hydroxyl group. All right, you got your carbonyl group here, which are either acetones or aldehydes. Right, so here is a ketone. A ketone I like to think of as a closed-in double bonded oxygen. Notice you got 2 carbons on either side of the double bonded oxygen. It's acetone, right? And that's a ketone, right? Notice it ends in tone, right? So that tells you that it's a ketone. An aldehyde is when you have a carbon double bonded to a hydrogen on the end of a molecule. I used to mix up aldehydes with carboxyl groups because a carboxyl group is an OH here, right? So you got to be really careful. This is the one mistake I used to make. I used to mix up these two. One is a carbonyl group or an aldehyde. The other one is a carboxyl group because it has an OH versus a hydrogen. This particular one in solution tends to give up its proton, right? And so they're often called acids or carboxylic acids is how I always remember them, organic acids. This is acetic acid. So notice the difference here, right? So acetic acid it has 2 carbons, acetone has 2 carbons. The tone tells you the ketone in the middle, or the acetic acid tells you have a carboxyl group or a carboxylic acid group on the end, like fatty acids do, which I showed you in the last slide, right? And so they give up the proton and therefore are an acid in solution. And if I back up one slide, right? Sorry, wrong way. Or 2 slides, I called this an acid because here is our carboxylic acid group or a carboxyl group, right, with the OH. And I told you it gives up that hydrogen, and so they're called fatty acids because they give up protons in solution. Sorry, going the wrong way. Oops. Amino groups are basic. Here's the amino group. It's an NH2. Here's glycine. All right, glycine is an amino acid. All right, so you can see glycine here. It's got an acidic group here, carboxylic acid group, and an amino group. So it's got an acid end and an amino end. All right, and when you have that, that's a specialized molecule. All amino acids look like this. All amino acids, as we're going to learn next time, are basically a carbon and a hydrogen, and then they have a amino group, right? And then they have a carboxyl group, right? All right. And so they got an acidic end and a basic end. Remember, the amino groups, you can basically absorb a proton from solution and make it more basic. And then this other group down here is called an R group. All right, and R groups can vary. All right, so they can be carbon-based, they can be nitrogen-based. And the R group determines kind of the amino acid. So there are like 26 R groups that you find in amino acids. Therefore, there are 26 amino acids, right? Because each one has a different R group. The R groups are going to help determine the function of the protein. Because when I start linking all of these amino acids together to form a really, really long chain, you're going to have R groups sticking out, right? Some of those R groups are going to be hydrophilic. Some of those R groups are going to be, for example, hydrophobic. And it's going to cause the protein, that long chain of amino acids called a polypeptide, right, to fold up into a particular three-dimensional shape. That particular three-dimensional shape determines the function of the protein. Form equals function. A really, really important concept in biology. Form equals function. So the sequence of amino acids in their R groups determines the shape of the protein, which determines its function. Again, a little more foreshadowing. All right, we're going to see that. Sulfohydryl groups and SH group here. These are really important. Here's cysteine. Again, another amino acid. How do I know that? Amino end, carboxyl end, hydrogen, carbon. This is the R group. All right? That's the R group there. All right? On glycine, guess what the R group is? On glycine, it's the proton is the R group. Glycine is the smallest amino acid. So that's the R group here. This is the R group here. Notice they're different, right? But they're both amino acids. Cysteine's pretty interesting. It has this SH group. This SH group can form disulfide bonds, all right, in certain molecules, all right? So disulfide bonds, as I'll show you when we get to proteins. All right, a disulfide bond is a covalent bond. And these covalent bonds are going to allow proteins to be very strong. If you have disulfide linkages between the amino acids, it'll help the three-dimensional shape hold its shape in solution. So these disulfide bonds become really important, like in cysteine. And this is called a thiol group. This SH group here is called a thiol group. Phosphates. You guys have probably heard of phosphates before, right? Phosphates become really important because they are going to form part of the backbone in DNA and RNA, and they are going to form the energy molecule we call ATP, right? So important with ATP and important with RNA and DNA, all right? And so phosphates are negatively charged, so they are polar, right? So they can dissolve in solution. And they also will be really important in holding energy in their chemical bond to in ATP. And I will show you how later on. The last group here, methyl group. Methyl groups are really important. They're added to a lot of different types of molecules. A methyl is a C with 3 protons on it. Methyl, remember, means 1 carbon. So these are your most common of your functional groups. Often they're called R groups, right, as I mentioned. And they help determine the function of your macromolecules. So my last slide here for this particular chapter is this one here, which shows you the importance of R groups in shape, right? Like I've kind of been trying to talk about, right? So form, as I mentioned, equals function. It's a dogma, all right? It's a very important theme, if you will, within biology. All right, so form equals function. As you can see here, we have male lizard, female lizard. I love lizards. I study reptiles. Male lion, female lion, right? These are the 2 molecules that determine sex, right? So we have testosterone and estradiol. They're both carbon-based organic molecules that are made from cholesterol, all right? So this molecule, this 4-ring structure, right? So A, B, C, and D, right? This is cholesterol, all right? You guys have all heard of cholesterol because there's good cholesterol and bad cholesterol. You actually don't need to ingest any cholesterol. Your body can build it. And what cholesterol is important in is it's part of the cell membrane, as we'll talk about when we get to membranes, but it also forms all the steroids. Estradiol, or what we call estrogen, and testosterone are the sex hormones, right? What I want you to notice between these two is look at them. They look very, very similar, don't they? They look very, very similar. You can see here we got this functional group that's different than this one, right? And then over here with estradiol, we have this hydroxyl group, and we don't have one there, right? The absence of that hydroxyl group there. That's, the major differences. And then notice your double bonds are in different places. And you would think, oh my gosh, those look really, really similar to each other. I mean, but look at the differences they cause. Major differences between male and femaleness, right? Major, major differences. And the molecules are really, really similar. As a matter of fact, you can convert testosterone into estradiol really easily by adding double bonds in this hydroxyl group. And there's an enzyme that does that. It's called aromatase because it cyclizes the double bonds, all right? And aromatase is obviously the enzyme that's really, really common in the ovaries of females because females produce testosterone first in the ovary, and then it's converted to estradiol. Then there are different forms of testosterone, like you got and estrogens. You got estrone, estradiol, estrogen. You got testosterone. You got dehydroxytestosterone, DHT. There are a whole bunch of different types. But it's the three-dimensional shape, and these R groups, either having different R groups or lacking them, that allow them to fit their receptor. Testosterone doesn't fit the estrogen receptor, all right? And estrogen doesn't fit the testosterone receptor because of the R group. The R group, it determines the three-dimensional shape. So functional groups and how they are arranged are really important in physiology and how life works in biochemistry and cell biology. Okay, wonderful. So that's where we'll end today. Next time, we'll now apply the carbon kind of characteristics and our understanding and foundation of chemistry and carbon to our macromolecules. Have a great day.